Yeah, am I audible? I hope I'm audible. Can anyone please confirm? Okay, so this is a 8-week course, and Final exams will also be MCQ. Whatever you're getting in your assignment problems, it's a similar set of questions and a similar pattern as well. Okay, I think we can get started. Yeah, welcome to week 4 of Advances in Omics. So, for the last 3 sessions, we have mostly focused on different types of sequencing technologies, the chemistry behind it, what kind of reactions happen during the course of the sequencing experiment, and how you get the signal and then you convert it into uh, a FASTQ file or a read file. And then we have also seen a little bit on the fact okay, how you can analyze the data, right? So today we are going to be mostly focusing on different types of sequencing projects. So population genetics level projects, and also what insights we can gain from that, and also like different types of uh, like biological events that we can interpret from sequencing data, namely evolutionary concepts or mutations which are causing some sort of selection and so on and so forth. So we need a lot of data for that, but the idea is that if you gather enough samples and sequence those samples, you can have an idea about a lot of different things, be it in humans, be it in different species, and so on and so forth. So we are going to start off with the concept of pseudogenes, right? So what is a pseudogene? Pseudogene is, if you define by definition, is a gene which is duplicated from an original gene, but the pseudogene does not have effective transcription power on it. So basically, the original gene will create the transcripts, it will create the proteins. The pseudogene will not be able to do the same. Because although the sequences are very similar, there will be very specific differences in very specific locations of the genes, which will like differentiate the original active gene with the pseudogene, and then the pseudogene will not be able to create the transcripts or the proteins, or at least the functional copies of that. It can create some defective copies, but those will not be used up. So there are a lot of pseudogenes in humans. There can be, say, for example, SMN1 versus SMN2. SMN2 is a pseudogene of SMN1, and these genes differ by like the sequence of these two genes are differing by 7 or 8 nucleotides, and it's like a 500 to 800 base pair long sequence. So these are very similar sequence uh, uh, regions of the genome, which are part of the same gene. It's just that the gene has a functional copy and it has a non-functional copy. The non-functional copy will be the pseudogene. So they have I ideally, what has happened is basically at some evolutionary point, these genes were functional, the pseudogenes were functional. Then mutations got accumulated in these genes and made it non-functional. And yeah, generally that is the mechanism by how it works. So they will not be able to code for proteins, but they can serve as molecular fossil records for evolutionary history. So, for example, if you have pseudogenes that are shared between multiple species, you can get an idea about the lineage of evolution of these species. Okay, so how can these pseudogenes be formed? So, as I said, at some point of evolution, there were some mutations that got accumulated in the gene, which made it a non-functional copy, right? So there can be many ways through which these mutations can occur and different types of mutations. Excuse me. So it can be that the promoter got missing, it got deleted. It can maybe the start codon got mutated. Maybe it had frameshift mutations. It had premature stop codons, missing introns, or a part of the gene or some exons are fully deleted. So a variety of reasons can be there for pseudogenization, as we call, converting full uh, uh, functional gene into a pseudogene. So a lot of these pseudogenes actually are located in chromosome 16, if I'm not wrong. And these are very near to your known hemoglobinopathy genes, HBB, HB1, HBB, and HGD, and genes like that. And along that region, there are a lot of olfactory receptor genes. So this region of the human genome is very prone to mutations, and it's a very highly selective locus. Right? So these pseudogenes and a lot of these olfactory genes you'll find in humans are actually pseudogenes. These are not functional genes at all. And if you see the evolutionary history of other species along with humans, you see that so humans are most closest to chimpanzees, and if you move away and away from the lineage, the number of pseudogenes actually increase. And like if so, for example, African elephant will have has the largest number of these olfactory genes, and a lot of these genes have become pseudogenes in us. So as evolution has progressed, these genes have accumulated mutations, and they have been converted from a functional gene to a non-functional gene. But why has this happened? Because as we have evolved, the sense of the requirement of strong smell apparatus or genes that will help you like kind of interpret the smell or like help in olfaction in general, it has reduced because we have moved away from these wild habitats. We have come into civilization and so on and so forth. So basically, we do not need these so strong olfactory receptors anymore. So these genes have accumulated mutations, and these have continued to persist in the human population. But yeah, we do not need them, and definitely these are pseudogenes, so this cannot function as well. Okay, so generally this is how pseudogenization happens, right? So another way to change the genetic dose, you can say, or in terms of the functionality of the gene. Another way to change is whole genome duplication. So basically, as by the name, it's how it sounds. It is going to fully duplicate a part of the genome or even the whole genome. Right. So one example of that is the evolution of teleost fishes, and these are the most species-rich vertebrate clad in the whole of animal kingdom, right? And so there is now solid evidence that has been found that there has been a whole genome duplication event in a recent history or at a common ancestor level of these extant teloids. And this is known as teloid specific whole gene duplication. And after this duplication, duplicate genes have had different fates because you're duplicating the same gene multiple times. Many things can happen with the duplicated gene. It can preserve the duplicated gene, it can remove the duplicated gene, it can preserve part of the duplicated gene, depending on the context and the evolutionary requirement. It can act like that, right? So the most common functionality would be to to have basically make it non-functional. That you have one copy of the active gene. Why do you need another copy? Because it'll have dosage issues and there will be other issues as well. So basically, in most cases, through evolution, these are made non-functional due to accumulating mutations. But in some cases, subfunctionalization can happen. There will be a partition between the ancestral gene and on the duplicate. The duplicate will have some subfunctionalization of the original gene, but it will not replicate the whole original gene in itself, right? So there can be many combinations of that. We'll come to that in a little bit later slide. But yeah, so uh, events like these can happen in the genome, right? And you can determine these events using sequencing data across different individuals. I'm talking from a human perspective. You can do it for different species, for different species as well. So you gather these human DNA sequences and you see that what kind of mutations have accumulated in the human DNA compared to, say, our closest ancestor, which would be somewhere between a chimpanzee and some other ape, right? So you can compare the whole DNA sequence of the decent ancestor with humans, and you can find what DNA changes have occurred, what kind of genes have become non-functional, pseudogenization, if there has been any duplication of any region of the genome that has happened, and so on and so forth. So basically, this omics data will help you kind of answer these questions in terms of evolution, in terms of functionality of the gene, in terms of if the mutations are under any selective pressure or not. So all these things could be answered using sequence data and omics data, right? So for humans, how we generally do it is using population genomics projects. So one such PopGen project is 1KGP, or the 1000 Genome Project, and a little bit of that I'm going to discuss. So this 1000 Genome Project came about very near to the end of the Human Genome Project. So it's like like early 2010s or like even before that, like late 2000s, right? In that time period. So what they try to do is get a set of samples. Initially, it was not 1000, it was less than that. Now it has gone above 1000. I think the 1000 Genome Project stands at around 2500, 2300 samples right now. But initially, what they had thought is they're going to collect 1000 genomes from across the world, across different ethnicities, across different populations, and try and sequence them and try to find mutations which are common across all the populations, which are different across all the populations, how the frequencies are differing, all these things, and if all these are actually translating to, say, for example, differential disease risk or uh, yeah, things like that. Basically, what kind of variations we see in the human genome and how these variations differ from ancestry to ancestry, population to population, geographic location to geographic location. So this was one of the major milestone projects of human genetics in the past 20, 30 years. And after this, there has been a lot of population genetic projects. One of them also currently happening in India, which is known as Genome India project. Also, they are doing collecting 10,000 samples from all over the country, and they're trying to see what kind of similarity or differences we have within the country also. So yeah, so a lot of population genetic projects have come up after this, but this was the first one. And some key takeaways, like some interesting finding from these projects, include selection signatures in certain genomic regions. So it detected, so there's the LCT locus, which controls for uh, lactose tolerance or intolerance in human beings. And there was seen that certain mutations in this LCT locus is associated with tolerance of high dose of lactose, right? So, and it was seen that these genomic regions which had this LCT locus had a high positive selection for these mutations, which help uh, tolerate the lactose were positively selected in European populations compared to other populations. So things like these small mutations that are giving physical characteristics to individuals are under active selection uh, in evolution. And since we have since there is a geographical barrier as well, and also there is a cultural barrier, and there are a lot of other societal factors like endogamy, consanguinity, things like this. So what happens is we form this cluster of individuals in a particular population which are kind of homogeneous among each other. So basically, in one population, there will not be much diversity. But if you compare that population to another population, there will be a lot of diversity because there has no intermixing happened between these two populations. But inside the populations, there has been a lot of endogamy, right? So uh, yeah, so we see, and at the current stage, we see a lot of these variations which are uh, which have different frequencies in different populations, right? And that is due to, as I said, a lot of factors, and the LCT locus is among them, and this study kind of brought that out in a more definitive manner. Uh, we also saw some so if you do principal component analysis on the genotype data, and if you do something called admixture analysis, I'm not going to go into detail, but if you cluster individuals based on their genotypes across all the SNPs that you have genotyped, and if you cluster them based on their zygosity of individual genotypes, so you find clusters of population on a PCA plot. So I'm just going to go here. Oh, I don't think that. Just let me. KGP. Um, yeah, so this is the latest one. This is the 2022 paper. Yeah, this I think has from 2200 to 2500, as I was saying, 2500 individuals. But just let me try to find the first paper. This is the latest one, I think. Yeah, yeah, this is the 2010 and the 2012 papers. The first one. So yeah, so this is actually the first paper of 1000 genome project, and if I go, I think I might have done some PC on this. Second, um, no, these guys probably not have. Second paper probably. Oh, oh wait a second. These guys also don't have PC. Uh, yeah, this is called an admixture plot. I'm coming to the admixture part. I'm just trying to see where the PCA plot is. Let me. Hmm. There's one paper for just the PC on 1 kg. Yeah, let's it is. Yeah, you can see. So this is a principal component, and so I'm not sure how many of you are all of you familiar what is a principal component analysis? Like, do you have any clue about what it is, or like any abstract idea, if not the mathematical base? Okay, okay. Uh, okay. Okay, I'll try to explain. I'm not going to go into the mathematical details. So what is PCA in a very layman term, what you can think is, so for example, imagine you have a multi-dimensional data, right? What do you mean by multi-dimensional data? That there are many uh, factors on the data. Say, for example, you have a RNA-seq data. So how does the RNA-seq data look? So it's a text data, it's a tabular data form, right? So on the first column, you have the set of genes. The first row will have each of the sample numbers, and and each box will tell you that this gene has this expression in this sample, right? It's a quite simple data. So you have one gene, you have multiple samples, you have gene expression values for all those samples for that gene, and like that you have, say for example, 20,000 genes. So if you have, say for example, you have done 100 samples, so your data would be, you have 20,000 rows and 1,000 columns. So 20,000 times 1,000 is your dimension of the data, right? So that is the dimension of the data. Now, since it has so many dimensions, it has so many columns, like so many factors in the data, you cannot visualize this data in two dimension, right? Because there is not one X, one Y that you can visualize X versus Y, right? Uh, there are like multiple X and there are multiple Ys, right? So how do you visualize that, or how do you reduce the dimension of the data? There comes principal component analysis. So we use eigenvalues and eigenvectors. If you have taken some linear algebra course or any matrix or determinant course, you'll know what eigenvalue, eigenvectors are. But generally, you take the eigenvalue and the eigenvectors, and using that, you reduce the dimension of like 20,000 by 1,000 into a representative two-dimensional data, X versus Y. So you collapse all the Y's into one Y, and you collapse all the X into one X, but preserving the variation among all these things. So basically, principal component analysis allows you to visualize higher multidimensional data in two dimension. In this case, you have, these are the two dimensions. This is PC1 versus PC2. That is how, so you will have, like, if your dimension of the data is n, you will have n PCs, principal components. But um, if you plot a PC1 versus PC2, or you compare principal component 1 versus principal component 2, it will show you the spread of the data and show you the variance of the data. So generally, that is how PCA works. Okay, like, is that helping you a little bit to understand? Like, have you gotten a little bit idea about what PCA is? Yeah. Okay. If anybody has any questions, any questions to ask, please stop me and ask. Okay. So now this is how the PCA will look like. Now, if in this case, what is the data? The data is a multidimensional data of genotypes. So what you have in your first column, you'll have all those samples, whatever number of samples. In this case, 1,000 samples you can issue. You have 1,000 samples, and in each column will represent each polymorphism, each mutation, each SNP, each indel, whatever you have called. Let us assume you have only called single nucleotides. So on an average, across 1,000 individuals, you'll have, say, for example, I'm roughly guessing, say, 20 million distinct SNPs. Now, all the individuals will not carry those 20 million SNPs. It's a union set across all the 1,000 individuals, right? So all the individuals will not carry all the 20 million, and not all the individuals will carry the whatever SNPs they are carrying in the same genotype. So first individual for the first SNP might have a heterozygous genotype. The second individual for the first SNP might have a homozygous, or a 0, 0 as a ref site. There is no mutation at all. That can also happen. So here, that is the structure of the data, and then you are collapsing the data into uh, two dimensions using PCA, and then you are visualizing it, the variation across principal component 1 and 2. And you can see the spread of the data, like what is the variation in the data. So you can see, so all these dots that you are seeing, what every mark on this is a sample, and these are colored, and basically, or the legend will tell you, depending on the color, depending on the shape, what population that it is coming from. And so basically, the colors are telling you what project. So these are different projects: HDP, PSNP, and 1 KGP. These are different projects, and these are so they have clubbed the three data sets from the three projects, and what they're doing is this. seeing the spread of the data across two-dimensional space, and then marking it by the ancestry of the individuals. And as you can see, similar ancestral individuals are clubbing together. So all these squares are African individuals. They've come from different data sets, but all these African individuals are clustering together. Here, all these are, I think, South Asian. These plus ones are South Asians. All these South Asian individuals are clustering together. And also, you notice how far the South Asian individuals are from the African individuals. So this is telling you relative diversity, relative variance in the data. Okay, all these individuals, even if they are carrying roughly the same amount of mutations, the mutations inherently are different, and also uh, the similar type of individuals will carry similar type of mutations. So that is all that you can get from PCA. So this is what is known as principal component analysis for genotype data. Generally, uh, if you have a population genetic data set, uh, the first thing you do is PCA. So all this 1 kgp thing that I'm saying, all the 1 kgp uh, data is available. You just go to data here. You can get the variants, you can get the data and everything. You just go to the FTP site and you can find all the data given one. Phase 1, phase 2, phase 3. In phase 3 also, what are the sequences, and in the data you get, these are the sample numbers. HG002 is the first, I think, sample. You can just click on any one and it'll give you all the data for that alignment, BAM files. If you go back, the exome we don't need. Read files also, FASTQ files also you'll get. You can do variant calling on that. You can merge the data and you can just create your own data set from here as well. So if anybody of you are interested, uh, please look into this. Sorry, yeah, this website for Thousand Genome Project. And if you want to play around a little bit, uh, definitely try. Sorry, can definitely try it, and you can let me know if you face any issues or if you need any help or anything. Since I cannot cover hands-on in this session, I do not have enough time or the scope to do it. If you want, uh, like if you are doing this and you are facing any issues or any problems, just let me know. I can try to help you out. Anyway, so that was PCA, and also yeah, how you generally get outcomes from population genetic databases. So there are a couple of other examples also I've put. So for example, there is this EPAS1 gene. So what happens is basically, if I remember correctly, I might be slightly wrong. You can just look it up. So the EPAS gene, one gene is involved in uh, hypoxia related conditions, right? Low oxygen content of the of individuals. It can detect and it can like trigger certain processes in the body that can counteract hypoxia conditions. But in individuals of Tibet, so Tibet is in the foothills of the Himalaya, right? It's in a very high altitude area. So there the uh, amount of oxygen is also less, right? So there the individuals have to survive in some low amount of oxygen. And it is seen that this particular gene, EPAS1 gene, has a mutation which actually kind of, it's not a loss of function mutation, but it is a mutation that does not allow the gene to uh, correctly estimate the hypoxic condition inside your body, right? And it is seen that this particular mutation is very well chosen, as in like it is very frequent in Tibetan individuals, because this has been positively selected generation after generation for these individuals to survive in this high heights without dying of hypoxia. So this is also something interesting. There's a lot of things that have come out in terms of if you see sickle cell anemia versus malaria, it's a similar story there as well. So in sickle cell anemia, you have this your RBC form sickle-like shapes, and it reduces the surface area of the RBC, and it cannot carry hemoglobin properly, right? Now, again, the RBC is also the home or basically the host of parasites like Plasmodium, which cause malaria, right? So if you have sickle cell anemia, you have a smaller surface area on your RBC. So you have anemia because the hemoglobin is not being carried by these sickle celled RBCs, and also Plasmodium cannot infect the sickle cell RBCs because it does not have enough surface area. So, uh, having sickle cell anemia gives you protection against malaria. So, these kind of selections, so this selection happens very frequently in individuals living in very dense jungle conditions or forest conditions where there is a lot of prevalence of mosquito-borne diseases. So there you'll see this mutation selected positively a lot. Even in in certain parts of India, certain tribal populations which who are very uh, like would live in this dense forest conditions or wild conditions, you'll find this mutation to be very common among them. So all this information so what you're getting out of this information is how the locus has been selected in terms of evolution, how the biology is working hand in hand also. So all this information you're getting from this large scale omics projects, this is genomics, but you can get some other answers if you do transcriptomics or proteomics, and studies like these are being done very commonly now. So yeah, just to I have kind of taken up a lot of time on this, but just wanted to give you an idea about what kind of things population genetics data can give you and what kind of insights it can give you. And I myself is also am also working in population genetics. I am part of the Genome India project, and there also we are seeing a lot of these things in the Indian population. Uh, and yeah, we're just so this paper is not yet published yet. It is in archive. So if anybody interested in population genetics of Indian individuals, maybe go through this. Okay, moving on. Okay. Yeah, as I was saying, there's a lot of variation in individuals. So these are all, so on an average, an individual will carry 4 to 5 million small variants. On an average, across all individuals, you'll find around 4 to 5 million variations, right? And you can see that that number, 4 to 5 million, is actually quite varied. So these are, so all these all these three-letter codes are telling you the population code for these set of individuals, and you see the variation. So these are African populations. These are European populations. Some of them are so DB, and these are basically, I think, South Asian populations. There are like all all over the world you have populations here, right? And if you see the variation in just the number, there is nothing else, not the zygosity, not the type of SNPs or anything, just the number of variations. It is quite varied from individual to individual, and it is like similar type of individuals have kind of similar numbers. So that is also you can see from these kind of data sets. As I was saying, in terms of uh, evolutionary history, you can model these mutations because there is certain amount of, so the mutations are at a certain level random and also at a certain level not random, because these mutations are then from passed on from generation to generation, and on an average we also know from one generation to next generation how many new variations are passed on. So we can determine the age of these variations using probabilistic models, and from there also there we estimated that there was some so the humans originated in Africa, and there was some um, bottleneck event that happened, which was the out of Africa bottleneck event that was around 50 to 60,000 years ago, where humans came out of Africa and then kind of settled in different places of the world, right? So, and you can see that the European individuals have more prolonged bottlenecks compared to Africans, and you can see that there's the much lesser genetic diversity in European individuals compared to African individuals. So things like these, you can get these information from these large scale data sets. Okay, coming to another important aspect of population genetics is studying ancient genomes. So generally, why we need ancient genomes? Because we need to find out what differences in DNA has happened across all these years of evolution compared to the ancient genome. So that is the general idea and how much we have evolved, what kind of changes we are seeing, what kind of changes we can predict in the future as well. But this has to be, the genome has to be old enough, right? So I think this, if you classify this as an ancient genome, this is a 4,000-year-old Eskimo genome that was sequenced, and it was a hair sample that was sequenced, and this is the earliest quality genome of an ancient human, because DNA is not, well preserved in nature. DNA is very unstable; it gets destroyed. So it is very hard to get older DNA samples, even with fossils, even with other like all these archaeological sites that we find. We do not find DNA there because DNA is very unstable. It does not remain intact for so many years of time. But since this was an, I think, a very snow-clad area, a very like frosty area, I think maybe that is why the DNA was preserved, and it was sequenced in 2010. And it was sequenced at an average depth of 20x, covering 79% of the genome. And it was seen that this individual was very similar, closer to the Siberian populations. Uh, uh, and It also provided migration pattern insights into the Arctic about what was happening in terms of migration and settlement for about 5,000 years ago. And they could deduce that this individual had brown eyes, dry earwax allele, thick hair, all these things from the DNA of the individual. So that was also very interesting. So if you go back even back, even further back, so you can go back to the nearest ancestor of ours, which is the nearest ancestor of Homo sapiens is Neanderthals. So our DNA, the human DNA, Homo sapiens DNA, has Neanderthal uh, some sections of the Neanderthal genome integrated into our DNA. So that is also a certain level of proof that we have evolved from Neanderthal individuals. So again, this was also, I think, sequenced. And yeah, and it was confirmed that Neanderthals contributed up across 2% DNA to non-African humans. For African individuals, that and the Neanderthal DNA is slightly more compared to Europeans and other individuals, but there is a substantial amount of Neanderthal DNA in us, right? And there was another uh, recent ancestor, which is the Denisovan individuals. So this was also found in, I think, 2010. So David Reich published this, and this is like about 5% of the DNA of these Denisovan individuals were found in Malaysians and Australians and other individuals as well. And also, we found interbreeding evidences uh, between modern humans, Neanderthals, and Denisovans. So there existed a time period in history where all these three species coexisted at the same time and they intermixed within each other. So in current individuals also you can find evidences of this integration between modern humans, Neanderthals, and Denisovans. So all these insights can just all these very useful insights, you have to say, and very important insights we are getting from just stretches of DNA, just sequencing stretches of DNA and finding out all these insights. So that this is very powerful, right? Yeah. So I'm going to come to the events that kind of define the evolution and help us study it. So I've just touched upon it a little bit in terms of whole genome duplication. So there you have uh, the whole idea is that you have uh, whatever the number of copies is there in your DNA. For humans, it's diploid. For other, in some cases, it's triploid, tetraploid, whatever is your ploidy. You increase or decrease that ploidy, right? So for duplication, you increase that ploidy. So if it is 2, you become 4. If it is 4, you become 8, something like that, right? So uh, yeah, so similar to gene duplication, whole genome duplication is a process by which an organism's entire genetic information is copied once or multiple times, which is known as polyploidy. We see this very often in plants. In animals, it is not so common. When it is present, it generally what happens is like, so I think just, just let me verify before saying this. 1 second. Uh, Yeah. So Down syndrome, which is a very well-known genetic disorder, it is caused by the trisomy of chromosome 21. So chromosome 21 has an extra copy, so that causes disease. So generally, having uh, ploidy differences causes diseases in humans. Right? In plants, it is mostly benign, and if you have read about, I forgot, I think the endosperm inside of the seed, the endosperm has, I think, tetraploid. It's tetraploid in nature. I don't know if somebody is studying plants, they can correct me. I'm not sure, but I think the endosperm of the seed has polyploidy. I think it is tetraploid, if I'm not wrong. Okay. Yeah. So evolutionarily, it is very important. So this leads to massive changes in cellular structures. It changes gene expression. It increases cell size, and it also can cause gene loss, right? It can cause gene loss during diploidization, uh, and it will also definitely duplicate certain genes. So it can have both long-term and short-term evolutionary effects on an organism's fitness with respect to the natural environment. But if it is beneficial to the species in terms of its survival and its reproduction and all the other things that it is needed to live, then it gets selected and passed on from generation to generation. But if it is not, it will generally cause diseases, and the mutation will be negatively selected, and these individuals will die or like not be able to pass on their DNA to the next generations and so on and so forth, right? Okay, so this gene duplication also contributes to genome diversity. They can uh, yeah, so the duplicated genes can undergo neofunctionalization or subfunctionalization, like the same gene duplicated. Now the duplicated gene has some new function or some older function kind of modified to help its cause, something like that, like not the exact same one. So it can happen like that, and it can help the organisms to adapt to their current environment. And also, most importantly, from an evolutionary perspective, speciation can happen. So what can happen is like it can give rise to a new species altogether, right? Because the ploidy has changed the amount of DNA, the whole DNA structure, the whole content of DNA inside that organism has changed. So it can definitely give rise to new species. So it has been hypothesized that many polyploidization events created new species via gain of adaptive traits or by sexual incompatibility with their diploid counterparts. So yeah, and one example would be the recent speciation of Allopolyploid spartina. It is a polyploid plant, and it is so successful that it is listed as an invasive species in many regions. So it is basically it it was generated because of polyploidization, but now it has become a species on its own. So things like this can also happen, which is evolutionarily very interesting to note. uh, yeah. So there is something called the 2R hypothesis. Sorry. Yeah. There is something called the 2R hypothesis. Uh, so there's something called Ono's original model. So this individual called Ono who gave this model, and he assumed that one round of whole genome duplication at the stage of fish or amphibians, and possibly another round of duplication between cephalochordates and urochordates. So these are all like different. There's a species belonging to different families and classes in the animal kingdom. So fish. So I think the course of evolution, if I'm not wrong, goes from reptiles, no, sorry, birds to reptiles to fish, if I'm not wrong, or like the other way around. Basically, they are connected, is what I'm trying to say. So, what one of the models says that like how human beings actually came into being and the vertebrates came into being is basically there are two duplication events that occurred, one here and the other one here. So both of them around like I think 500 to 600 million years ago. That is the assumption. This is the 2R hypothesis. Uh, the there is something called 3R hypothesis as well, where there is a third uh, location where the duplication has occurred between these two set of species, and uh, yeah, and this is a fish-specific whole genome duplication. And survey sequencing of the elephant shark genome showed that it's four Hox clusters like mammals, indicating 1R and 2R preceded the radiation of the jawed vertebrates. So basically, elephant shark is not a jawed vertebrate. We are a jawed vertebrate. Mammals are jawed vertebrates, right? So, and you can see these Hox genes. Hox genes are very important. These are homeotic genes. These help in during embryo formation and basically zygote to the embryo, from zygote to the embryo, and how differentiation inside that occurs. So Hox genes are very important in that. So and that Hox gene region is conserved between us and elephant sharks, which kind of tells you that the jaw vertebrate thing, the jawed thing that differentiates us with elephant sharks, that thing happened after uh, uh, the uh, yeah, after the duplication event, because if the duplication event had uh, happened after the jawed differentiation had occurred, then we would not see these Hawks similarities between us and some elephant sharks. Okay, yeah, and yeah, so these are uh, different levels of functionalization and that can happen with respect to whole genome duplication. So I think uh, this image itself is kind of a lot intuitive. So what happens is you have a singleton gene, then you have duplicated gene. This gene is duplicated into two. Then what can happen is hypofunctionalization. So some part of the work of that gene would be taken up by the first copy. The second part would be taken up by the second copy. So this width is sorry, the length is reduced or width is reduced. Whatever you can say. Right. Subfunctionalization is you have in hyperfunctionalization, it will reduce the expression of both the copies, right? If I don't know if you're aware about X-linked inactivation in human females, right? So human males have one copy of X, one copy of Y, and human females have two copies of X, right? Now, the gene expression of the genes of X chromosome in males, because there is one copy, will be lower if you compare it to two copies in females. But then there will be like gender differences in terms of the biology itself. But that does not happen. What happens is there's something called X-linked inactivation, where one copy of X in females is suppressed, so it does not do any kind of functionality. It only functions by one single copy, which males also do. So that is how that. So there's a reduction in one of the copies, a reduction in expression in one of the copies of X in human females. Similar type of a thing is happening here as well. This is hypofunctionalization. Basically, both the original and the duplicated copy will have reduced expression levels to kind of match the original single copy level, right? So that is hyperfunctionalization. In subfunctionalization, the division of the function happens such that two members of a pair confer only of part of the only part of the functions of the progenitor singleton. So basically, it divides up the work. So you will do this. One gene will do this part of the work. The second gene will do another part of the work. New functionalization, as I said, happens because of like happens that the duplicated gene becomes has like a separate function altogether. It is not similar to the original gene. Right. Yeah. So yeah, that is new functionalization. Yeah. In dosage balance, what can happen is it can mediate retention of specific classes of genes that are dosage sensitive, like the X-linked inactivation I was just talking about, to maintain their stoichiometric relationship with partners in multi-component interactions. So basically, if you have some dosage sensitive genes here, if you if you overexpress those genes, there will be some like pathogenic phenotype or a disease-causing phenotype or something like that. So what happens is basically they reduce the dosage, the expression of each of these set of genes in specific locations, so that to counteract this phenotype. Right. Okay. Yeah. And in compensatory drift, the duplicates can diverge depending on the total expression. A dosage balance effect will still operate with their partners. So what will happen is one will become too high, another will become too low. So if you average it out, it will become the same. So that is compensatory drift. And neutral variation, as the name suggests, that different members of a duplicate pair might have different expression levels under certain conditions in which dosage or function is not an object of selection, similarly to apparent neutral variation of singleton gene. So basically, this will not cause any issues. This will not cause any evolutionary pressure. This will not give a different phenotype or anything else. So this is just neutrally, it is there. It is maybe changing the dosage, but it is not causing any issues or it is not causing any phenotype. Okay. Yeah, and basically, all this rounds up to what is selection and what is in terms of evolution. So, natural selection, as we all know, is a differential survival and reproduction of individuals due to differences in phenotype. It is a key mechanism of evolution, the change in heritable traits characteristic of a population over generations. And Darwin popularized this term of natural selection, contrasting it with artificial selection as well. So yeah, we all know what natural selection is. So all these things, all these duplication events, or even duplication events, or changing of, like, so integration of Neanderthals, or say for example pseudogenization, all these are kind of contributing to the evolution, right? Because all these are introducing the DNA mutations or different DNA inside these organisms, and these organisms through generations are positively or negatively selected by evolution. Okay. Yeah. Now how, like, as I said, natural selection will happen because there will be DNA changes, mutations, and so on and so forth. But how do you quantify it? How do you quantify that there is some positive selection happening on this gene, or some negative selection happening on this gene, or it is neutral? Right? So there are a lot of mathematical techniques to do that. One of them is a very simple one, which is the Ka by Ks or Dn by ds method we know. So basically, this is a statistic that compare rates of non-synonymous substitutions to synonymous substitutions, right? I am not sure if you're aware of what is non-synonymous and synonymous. So non-synonymous substitution it's a mutation. Both of them are mutations. Both of them are single nucleotide mutations. So in synonymous substitutions, what will happen is even after the mutation of substitution of, say, for example, a T to C has happened or a G to A has happened, something like that, even after that mutation, the codon remains the same and the protein remains the same. So even after the mutation has occurred, there is no change in the actual expression of the transcript or expression of the protein, because it's the same thing, because just the codon has not changed. The codon is the same. The amino acid coded for that particular changed nucleotide is also exactly the same as before. So that is synonymous. Non-synonymous are variations like missense variations. Uh, the classic sickle cell anemia variant, which is glutamic acid to valine, if I'm not wrong. Yeah, glutamic acid to valine in codon 6, if I'm not wrong, in the HBB gene. Like it's a missense variation. There, the amino acid is changing from glutamic acid to valine, right? So amino acid change is happening, so that is non-synonymous. If no amino acid change is happening, it is silent, it is synonymous. So if you find the number of non-synonymous variants in a generation versus the number of synonymous variations in a generation, and you find a ratio between them, that will tell you in which direction the evolution is moving. Are you accumulating more non-synonymous variations, or are you accumulating more synonymous variations? So the idea is that a relative excess of non-synonymous substitutions indicates ongoing positive selection, favoring novel protein structures. And so basically, if your value dn by ds or ka by ks, however you want to call it, is greater than one, uh, will be positive selection, whereas smaller values indicate ongoing negative selection against deleterious mutations, because these missense variations or these non-synonymous variations, these are to a certain level deleterious in nature, right? Because they're changing the protein, they're changing, sometimes truncating the protein, and so on, right? So yeah, so if it is positive, sorry, if it is greater than one, it is positively selected. If it is negative one, then sorry, less than one, then negative selection is happening. Now, there are various methods to calculate this ratio, like various models for calculating synonymous and non-synonymous substitution rate, and they take into account different probabilities of different mutations. Say, for example, what do I mean by this is, for example, transitions are more likely than transitions. So a purine to purine change or a pyrimidine to pyrimidine change is a much higher occurrence than a purine to pyrimidine change, right? So all these factors you can accumulate and make a probabilistic model. So this is how uh, uh, synonymous mutations should appear. This is how non-synonymous mutations should appear with respect to a null hypothesis. And then you see what is there in your data, and if it is deviating significantly, you you accept the alternate hypothesis and you see a statistical difference between them. Right? Okay, so one such model is Kimura's neutral theory of molecular evolution. Uh, and it says that the vast majority of genetic change is attributed to genetic drift rather than Darwinian selection. Right? What is genetic drift versus Darwinian selection? However, as researchers began to develop methods to distinguish neutral from adaptive changes in the genome, many came to reject the stronger versions of the neutral theory and turned their attention towards quantifying relative contributions of drift and selection to molecular evolution. Importantly, however, neutral theory enabled development of tests for selection by assisting sophistication of models of molecular models of genetic drift. In many tests of selection, we compare empirical data against data generated by simulations of drift. As I was saying, you have a model, probabilistic model, that is your null hypothesis. Then you compare it with your actual data and see if there are statistical differences among there or not. So yeah. So generally, uh, yeah, so these are different methods for determining macro evolutionary selection, right? So this is one part of the genome across four species, and you can see that species 3 and species 4 differ in the nucleotides quite a bit. And uh, yeah, and so these traits are conserved across many clades of phylogeny, but show an extreme differentiation in one or few lineages, and these are likely candidates for selection. So if you have very similar species having very similar sequences, but there is one difference, it can tell you that probably this region of the genome or the phenotype associated with this region of the genome is going towards selection, right? So, and then you do metrics, use metrics such as Ka by Ks or Dn by Ds, as I was saying, compare this rate of non-synonymous, which is amino acid altering, versus synonymous, which does not alter, change the amino acid. You can use this, and there are some couple of tests, which is firstly is the McDonald treatment test. We are going to, I think, talk a little bit about this, and also the Hudson treatment test. They hinge on the intuition that levels of interspecies divergence and of intraspecies polymorphisms are governed by mutation rates and are correlated unless selection of some other force is happening. So the idea is that the difference in genetic, uh, say for example, the yeah, the difference in genetic sequence between two species. So that divergence and also interspecies, the same species having different DNA sequences, both these things are governed by mutation rate, which is natural. Like if you go from one generation to another, there will always be some mutations that are additionally introduced in every generation. So that is the mutation rate, right? On an average, from father to son, how many mutations are carried forward, and what is the rate of new mutations that are coming, right? So both intraspecies divergence and interspecies divergence both are governed by mutation rates unless and until there is selection happening. So that is the whole hypothesis. That unless and until that mutation is giving you some benefit, right? If not, then it's just the mutation rate is at play. There is no selection happening. Yeah. Okay. Yeah. Just give me a moment. I'll be back in 2 minutes. I'm audible, right? It's a thumbs up or a yes would be fine. Thanks. Okay. Now we talk about a little bit about the tests or methods that we use to quantify this divergence. Right. So one of them is the MK test, which is the macronal treatment test. It's similar to what we have already discussed, that the ratio of your non-synonymous to synonymous mutations should be fixed within a species and also between species. So that the divergence interspecies and also interspecies with respect to non-synonymous and synonymous mutations, they need to be constant unless and until there is some selection at play here. Right. So deviation from this would imply that there is some selection going on. So this is how it is defined. So this is the synonymous set of mutations, non-synonymous, and for within species you give it as PS. For synonymous, for non-synonymous it is PN, and similarly between species DS and DM. So if the evolution is neutral, there is no selection happening here, then the ratios should almost be equal or very close to each other. If positive selection is happening, so you are finding excess of non-synonymous differences compared to synonymous mutations, then it is a signal that some sort of natural selection is at play here. And for purifying selections, uh, you will see that the number of non-synonymous mutations have gone down much more compared to the synonymous mutations. So there, some sort of negative selection is happening, some sort of purifying selection is happening where you are removing harmful mutations from the genome, right? So that in that case, PN by PS will be greater than DN by DS, right? So these three are the interpretations of MK test, and they basically measure alpha. It's same thing. It's just that we are using a different variable to measure this. It's the same thing. Alpha would be one minus this um, whole expression, and if it is greater than zero, it is adaptive evolution is happening. Positive selection equal to zero, close to zero is for neutrality, and if it is much lesser than zero, it is purifying selection or positive selection. Sorry, negative selection. Yeah. So this is the general NK test. This is the formula. Small problems might come in your exam related to this formula, so please keep in mind. Then we talk, I think, sorry. Then we talk about one of the like most widely used test statistics for quantifying these kind of issues is Tajima's D test. So what it does, it detects deviations from neutral evolution by comparing two genetic diversity factors. One is the pi, another is the theta or theta W. Pi is telling you what is the average pairwise number of nucleotide differences between two individuals or two species. Theta would be the expected diversity from the number of segregating sites. So this is a simulation. This is telling you what probabilistically what is the expected diversity, right? So this is the formula. You have pi, uh, you have theta, and you have root over variance of pi minus theta w, which gives you the d. So this d value, again, similarly, if you have uh, zero, close to zero, it is neutral. If you had greater than zero, you have excess of intermediate frequency alleles, basically balancing selection. Some sort of selection is happening, positive. And if t is less than zero, excess of rare alleles, purifying selection is happening, population expansion is happening. Like purifying selection is negative selection is happening. So, how would you calculate Tajima's D? One example is given here. So these are four segregating sites, four sites you can assume in across four different species. And these are the four nucleotide sequences. You have to count the positions where there are variable, uh, as in like differences in nucleotides. I think across all these four sequences, there are six. I can see one here, one is here, mm, two, this is two, this is three, um, and this is four, this is five, and last one six. Right? Where there are six variable positions across the sequences. So if you have uh, uh, the variable number of sequences, then you can calculate the theta w. The formula of it is given by S by AN. What is AN? AN is basically given by this term. It is uh, uh, infinite sum, not infinite. This is a finite sum of one to one, 1 to n minus 1, 1 by I. So that is your uh, uh, alpha, and the n would be the number of sequences. So for n equal to 4 sequences, alpha would be 1 plus half plus 1/3 because n equal to 4. So alpha will go from 1 to 3 sigma of 1 by I. So if you add this up, this becomes 1.83. And if you calculate the theta w would be S by AN, right? S is the number of segregating sites, which is 6, and divided by 1.833, which has come, which is the A value or the alpha value. So theta W would be 6 by 1.833, which is 3.27. Now you calculate pi, which is the average pairwise nucleotide difference. So pairwise difference across six pairs is 18. You can check between pairwise. So 1 versus 2, 2 versus 3, 3 versus 4, like this, how many differences do you have? If you calculate it, it will come as 18. So pi would be 18 by 6 because there are 18 differences and 6 pairs. So uh, pi is given by as average pairwise. So 18 differences across six pairs. So you find the average 18 by 6 equal to 3. And then d you can calculate from here directly because you have pi minus theta w. And since there is no variance in pi minus theta w, if you have a lot of data, then you calculate the variance. But here there is only one data point. So you just, this variance, this is one basically, right? So you just remove this pi minus theta w, 3 minus 3.27, which is point 0.7, negative 0.77. If it is negative, then purifying selection is happening, negative selection is happening, excess of rare alleles in the population, and population is expanding. So this kind of numerical, right, can very well come in your exams, in your assignments. So please practice this kind of examples. These are not very difficult problems to solve. You just need to know the formula. What is A? What is theta? What is S? From there, you can calculate theta. If you have theta, then you calculate pi. And if you have theta and pi, you have the formula, you calculate D. It's not very difficult, but you need to know the formulas and how to work it. Yeah, I think that's all in terms of my discussion. Before I start the questions, discussing the questions, if anybody of you have any doubts. Okay, if no questions, we'll move on to the assignment problems. Yeah, I have not kept the first question. Just let me go here. Yeah, so this was the first question. Yeah, I have an issue with this question, so that is why I have not kept it. So this is the first question, that one species is smaller in size than another species. This is statement one. So statement two is that the other species has one excess round of whole genome duplication compared to the first one, right? Then they're asking if what are the statements that are correct, what are the combinations that are correct and wrong. So here uh, the answer is both statement one and two are incorrect, right? So I'm not sure, like I myself am not familiar with this species. Last year, actually, we had a different answer for this, the same question. So this year, the answer has changed somehow. I'm not sure how and why it has changed. But my accepted answer would be that statement one is true, but statement two is not the correct explanation for statement one. So the second species has a whole genome duplication compared to the first one, but that does not necessarily mean that the second species will be bigger in size compared to the first one. The size of the species or the size of the animal does not matter here. So I am not agreeing with the answer that is given here. So that is why I have not put this question in the set that we were discussing. But still, I wanted to mention this question and discuss with you. So yeah, okay, moving on. Okay, these are questions that you need to know. Like as in like if you cannot work this out, cannot like logically think about it. If you know, you know. If you don't know, you don't know. Okay. So yeah, please put the answers in the chat box. I'll wait for these are easy questions. I'll wait probably 30 seconds for this. A couple of C and D. The answer is three. So yeah, there's something if you don't know, you don't know, but you should know this. So it has three successive WGD events. Uh, it's a highly polyploid organism. So answer is three. Yeah, that's correct. Common ancestor of humans and chimpanzees. Chimpanzees are not direct ancestors. Something in between. Chimpanzees are, you can say, our first cousins. So yeah, we are common ancestor of humans and chimpanzees. Yeah, FST is something that we have not discussed, but FST is very, very, very important in population genetics. So yeah, okay. The answer is B. It's not mutation rate. It's calculated with respect to the mutation rate, but it is not exactly the mutation rate. So I'll just quickly show you. FST is a very uh, yeah, HT minus HS by HT. HT is the expected heterozygosity of the total population. HS is the average expected heterozygosity, and yeah, expected versus observed due to, and basically it ranges from 0 to 1. So the closer it is to 1, the more different the two populations are. The closer it is to 0, the more similar the two populations are. So yeah, FST and I think Tajima is D to a certain extent, and also um, yeah, fixation indices like this actually calculate population differentiation. Okay. Sorry. Okay. I think it is ACD, I think. Yeah, ACD. Right. So the question is, which of the following is true regarding codon-based tests? So it is applicable to protein coding regions, which is well known. It is applicable to all types of nucleotide sequences. That is not correct because unique. to find protein coding regions of the genome and then um, like calculate. And it can be applied interspecies or interspecies among populations as well. So the second option is not correct because you need specific regions of the genome which code for proteins. So not all types of nucleotide sequences. Oh, I think it's Charlie frequency. Okay. Do we have any answers? B and D. Okay. Okay. So this would be A, B, and D. Yeah, A, B, and D. The thing is, it cannot be applied interspecies because the variations are not the same, right? So if you say that the variation and the frequency for one variant in humans is higher compared to in, say, for example, chimpanzees, it does not mean anything because the context is all different, right? Chimpanzees have that variant for some different reason. Humans have that variant for some different reasons. Calculating frequency of the variance and comparing between interspecies, it is not the correct way to do because you will not get anything because the contexts are widely different. It can be applied in species among populations, right? So, for example, European versus South Asian, what is the allele frequency difference for a particular set of variants? That makes sense, but not between humans and chimpanzees. So, yeah. Yes, that's correct. So, stop codon. So, which of these are stop codons? I think actually without TGG, everyone, UGG is not a stop. So basically, this will be U in RNA. So UGA is a stop codon, UAG is a stop codon, UAA is a stop codon. UGG is not a stop codon. So yeah, it's correct. Okay. Uh, the answer is Tajima's T and population differentiation because in Tajima's T you calculate what are the pairwise differences. There you're taking into account frequencies of these alleles across different sequences. And in FST also, you're definitely using the frequencies. So FST and Tajima's T is the correct answer. Moving on. Yeah, that's correct. You have to compare it to primates because you cannot just compare it to uh, a fish or something. There has to be some context behind it since primates are our closest ancestors. So, yeah, other primates, including chimpanzees and other apes, if you compare, you can, depending on the evolutionary distance between us and those species, we can have an idea about uh, how the evolution has happened. Yeah, this should be pretty easy. A, C, D. Why not B? What is wrong with B? Actually, the answer is everything can happen. Like non-functionalization can happen as well. Like you have a whole genome duplication, one of the copy of the gene is active, functional, the other copy is non-functional. Can happen, right? So all of these are possibilities. Right. Yeah. Okay. This should be also very easy. Okay. Yeah, that's correct. So generally, you do experiments on E. coli or other bacteria or Saccharomyces or organisms like this because they have shortened generation time and you can like I think E. coli has about 120 minutes or something like that. So you can generate them very fast and you can observe the differences from one generation to another. Okay. Mm, yeah. Okay. Mm, Hmm. Yeah. I think somebody said DK. Yeah. Yeah. Why would it eliminate the potential for further evolution? It's just that you're increasing the copies of your DNA, right? You're increasing the ploidy, you're increasing the amount of DNA, but it does not take away from the fact that it can mutate in the next generations and cause, like, it can undergo selection, right? So, what is stopping it from have undergoing evolution? Actually, nothing, right? So, the more mutations you accumulate, depending on the type of mutations and basically if it is uh, showing a phenotype or not, that will result in positive or negative selection, right? It won't depend on how much DNA is there. And genetic variation will not be lost because it's the same genetic variation you're copying, right? So it won't be lost. And chromosome number will definitely not be reduced because it's the same number of chromosomes, it's just increasing the amount of DNA. So the first option is the correct one, but it can generate new functions, gene functions, which is called neofunctionalization. Yeah, yeah. This, if you know, you know. If you don't know, yeah, you know, you should know. Yeah, this the answer is Chinese mudjack. It's a type of a beer. Yeah, it's type of deer species. It's not gorillas or it's not chimpanzees. It's type of deer species, and it has 46 in females as well. So, incidentally, the Indian mudjack, which is its close relative, it has 6 or 7 chromosomes. So it is quite fascinating. Okay, moving on. Okay. Yeah. So these are the two tests that rely on non-synonymous versus synonymous. The non-synonymous has to be present in the codon, otherwise that does not make sense, right? So, yeah, moving on. Yeah, this also, if you know, you know. The answer is protozoa. Uh, it has very extreme genome architecture, and they, in their lifespan, they fragment the DNA into millions of pieces, and these are known as nanochromosomes. So, yeah, this is very, very, very high number of chromosomes for protozoans. Moving on. Yeah, it's the same question that I had discussed here. It's the same 4 set of sequences. I This slide will be in here. So it's the same exact question there. So I'm not going to discuss there. Just yeah, just let me know if you find any issues solving these problems. I could not find a separate one, so that is why I took the one that is already given here. Yeah, so the answer here would be 3.278, 3.3. I think we got it there. It is negative, so I think it is the Tajima's D is negative 0.77. So it is not negative selection is happening. So reset selective soup. So this the answer to this would be C, and the answer to this would be 3.278, which we calculated earlier as well. So it's the same question. I'm not going to repeat. Okay. Yeah, this is maybe this is. Mhm. Yeah, Marsic is something that we have not discussed, but it is like discussed a little bit in the recorded lectures by Sir. So yeah, yeah. The answer is all of them are correct. So it generates pool tag deletion strains. It grows deletion pool in specified conditions. Then it purifies DNA and sequences the barcodes, and then it computes the average abundance of these barcodes across these pools. So all of them are correct. And the bar stands for barcode analysis by sequencing. So yeah, we have not discussed it in our session, but it is discussed in the live in the recorded lectures. So yeah, you should know this. And finally, this is the final question. Okay, wait for another 30 seconds to see if anyone else answers. Okay, no other answers. Yeah, I think B is not there. I think you answered B. Yeah, A and C are the answers. So the question was, uh, in a allele frequency-based test of selection, what can be the inferences affected by demographic events, mutation rate heterogeneity, GC bias gene conversion, and recombination rate variation? So demographic events will definitely change the change the inference because, like, in terms of, say, for example, in Tajima D, so a demographic event like a population found bottleneck or a founder event, it can mimic balancing selection across the whole genome, but actually it is not. This is a population bottleneck, right? And recombination, rare variations, and basically how and where the recombinations are happening will change the allele frequencies of the variations around this recombination blocks. So recombination rate of meiosis is not equal across different parts of the genome. There will be differences in different region, and if the recombination rate is high, the mutation rate can be higher. So that is not a signature of selection or evolution. It's the biology itself. It's how the meiosis is happening. Uh, and yeah, so uh, recombination rate variations can affect the linkage of the alleles, which two alleles would be inherited together versus which won't. And it can create these false signals of selection, but actually it's just the biology working itself. right? The mutation rate heterogeneity will not play a role because the mutation rate heterogeneity is telling you if the selection is happening or not. Right? And also GC biased gene conversion is also not related to selection tests. So A and D are the answers. Okay, so yeah, that's all from my side. If you have any questions, doubts, anything else to discuss, please go on. Okay, if there are no further questions, I'll stop the session. Thank you for joining. I'll see you next week.